Hi everybody! Today we're going to be talking about protein synthesis. The genetic code of DNA codes for proteins. Say you wanted to make some proteins that would help break down the food you had for lunch today. Somewhere in your DNA is a gene that specifies the instructions or blueprint for that protein. You might even have several copies of that gene if the protein is important enough. A gene is a section of DNA that carries the instructions for how to make a functional protein. To make the protein you want, we must first make a copy of the appropriate part of the DNA. We make a copy because we want to keep our DNA safe and secure in the nucleus. We don't want to expose it to anything that could damage it. That copy is called RNA. The RNA copy carries the gene's instructions to make the protein. The process of going from DNA to RNA to proteins is called gene expression. Because when you make the final product, the protein, you are expressing the directions coded for in the DNA. The order of operations, DNA to RNA to proteins, is called the central dogma of molecular biology and is conserved throughout life on Earth, meaning that almost all organisms do it the same way. Each process has a name. When you copy DNA to RNA, it's called transcription. When you make a protein from RNA, it's called translation. One way to remember this is that DNA and RNA are both nucleic acids. They both use the nucleotide monomers to store information. When you make a copy of DNA to RNA, you transcribe or make a similar copy. However, when you use RNA to make a protein, you are switching languages. You're switching from the language of RNA, which is nucleotides, to the language of protein, which is amino acids. You have to use a translator to speak the other language. Therefore, we call this translation. There are two differences you might notice between DNA and RNA. Look at the letters of RNA. What do you see? RNA has a U instead of a T. U stands for uracil, and in RNA it exists instead of thymine. The other difference is that RNA is single-stranded and DNA is double-stranded. In prokaryotes, there is no nucleus, so DNA is transcribed to RNA and the RNA is translated to proteins all in the cytoplasm. In eukaryotes, transcription of DNA to RNA happens in the nucleus, and then RNA leaves the nucleus and is translated into proteins in the cytoplasm of the cell. You can see in both types of cell that the ribosome is our translator for going from nucleic acids to amino acids. Let's start with transcription. This happens in the cytoplasm in prokaryotes and in the nucleus in eukaryotes. One strand of the DNA is used as a template. In DNA replication, we know that the enzyme responsible for making new DNA strands is DNA polymerase III. In the process of transcription, the enzyme responsible for copying the DNA and making new RNA is called RNA polymerase. You can remember it is called RNA polymerase because it's making RNA. RNA polymerase uses the template strand to synthesize the new RNA molecule. The other strand is called the coding strand. If we go back to this graphic, you can see the two blue strands of DNA. Imagine they are pulled apart, and RNA polymerase starts making a copy. The top strand is the template strand, so RNA polymerase will move along, allowing the correct bases to pair and catalyzing the bonds between the nucleotides. If you look at the resulting orange RNA molecule and compare it to the top blue NA strand, you will see it's complementary. Starting on the left, A in the DNA is complementary to the U of the RNA. C in the DNA is complementary to the G in RNA. C in the DNA is complementary to the next G in the RNA, and so on. This makes sense because we used the template strand to base pair with. Now take a look at the other DNA strand, the one on the bottom, and compare it to the RNA. With the exception of U's being there instead of T's, they are identical. The code in the RNA is the same as the code in the DNA. So the bottom blue strand is called the coding strand. It is what will determine the order and number of amino acids during translation. The other thing I'd like you to notice here is that the messenger RNA is arranged into a series of 3 nucleotides. Each set of 3 nucleotides are called codons. Each codon will specify 1 amino acid. If you figure out how many possible combinations there are, you have 4 nucleotides and 3 nucleotides per codon. That's 64 possible combinations. There are only 20 amino acids. Therefore, although each codon specifies a particular amino acid, there is some redundancy in the code. What this means is that more than one codon will specify a particular amino acid. The master genetic code chart correlates the codons to their amino acids. You can also see that there are 64 combinations of the 4 RNA nucleotide letters. The letters AUG are the start codon. This is the signal to the ribosome to start translation. AUG codes for the amino acid methionine. There are also 3 special stop codons. They don't code for any amino acid, but they do signal the ribosome to stop translating the RNA. We're going to talk about transcription in prokaryotes because it's simpler. There are 3 phases to transcription: initiation, elongation, and termination. Initiation begins when RNA polymerase recognizes and binds to a promoter on the DNA. The promoter is a sequence of DNA that signals the start of a gene. Next, RNA polymerase will unwind a short sequence of DNA, breaking the hydrogen bonds that hold the 2 strands together. You can see it forms a little bit of a bubble. We call this the transcription bubble. In stage 2, RNA polymerase will add new RNA nucleotides to the growing RNA molecule at the 3' end. The RNA will not stay bound to DNA for long. So as RNA polymerase moves along the template strand, the new RNA molecule trails away, and the DNA will reform its hydrogen bonds between its nitrogenous bases. When RNA polymerase reaches a sequence in the DNA called the termination sequence, it detaches from the DNA and releases the new RNA molecule. The RNA molecule is called a transcript, and in prokaryotes, it is ready to be used for translation right away. Transcription in eukaryotes is far more complicated. There are several different RNA polymerases, and other factors called transcription factors are required to help RNA polymerase bind to the promoter. The RNA must be processed before it can be translated. To process an RNA transcript in eukaryotes, first a cap is added to the 5' end, and a tail made of lots of adenine nucleotides is added to the 3' end. The cap helps the ribosome recognize the transcript, and the poly A tail helps to stabilize the transcript. In addition, the cell will cut out unneeded parts of the RNA. The unneeded parts are called introns, and they are shown in pink on your screen. The useful parts, or the parts that we keep, are shown in red, and they're called exons. Once we have stabilized the transcript and cut out the non-coding regions, now it's ready for translation. We just finished talking about transcription, or the production of RNA from DNA. Next, we're going to talk about translation, which is the process of using RNA to make protein. But first, we have to talk about the types of RNA. The one we'll spend the most time talking about is messenger RNA, or mRNA. This type of RNA is the code that is translated to make a protein. Another type of RNA, transfer RNA, or tRNA, is what does the translation between nucleotides and amino acids. The third type of RNA is called ribosomal RNA, or rRNA. This type of RNA is what makes up the ribosome. Neither tRNA nor ribosomal RNA are used to code for a protein, but both are very important in helping to make proteins. Remember, tRNA is made out of RNA nucleotides. At one end of the tRNA molecule, there is an anticodon. This is complementary to a codon on the messenger RNA. Remember, a codon is a group of 3 nucleotides. The tRNA will base pair with a specific codon. In this case, of this tRNA molecule, it will base pair with a codon that reads UUC. The other end of the molecule is a binding site for a specific amino acid. The tRNA will bind to its amino acid and bring that amino acid with it when it base pairs with the correct codon. Now you can see why we call tRNA our translator molecule. On one side, it can bind to nucleotides, and on the other side, it binds to proteins. The ribosome is made up of ribosomal RNA, or rRNA, and proteins. It has two subunits. The small subunit binds to the messenger RNA, and the large subunit will bind after the first tRNA arrives. Once assembled, the ribosome has three different sites for tRNAs. The A site is the acceptor site. The P site is where one amino acid is joined to the next with a peptide bond, and the E site is where the empty tRNA will exit the ribosome. The initiation of translation is shown in detail here. Messenger RNA binds to the small subunit of the ribosome. tRNA will bind to the start codon, AUG, through complementary base pairing. The large subunit will assemble on top, placing the initial tRNA at the P site of the ribosome. We are now ready to begin making the protein. The second phase of translation, elongation, is shown here. The ribosome is moving along the messenger RNA towards the right. A new tRNA enters the A site. The anticodon of the tRNA will bind to the messenger RNA. Only the correct anticodon will bind. The covalent bond holding the amino acid chain to the tRNA in the P site is broken, and a new covalent bond is made with the amino acid at the A site of the ribosome. The amino acid chain is now connected to the tRNA at the A site. However, the ribosome is still moving. As it moves along the DNA to the right, the tRNA that was in the P site gets shifted over to the E site. It no longer has an amino acid attached to it, and it's released. The tRNA that was in the A site, as the ribosome moves, gets shifted to the P site, and a new tRNA enters the A site. The cycle starts again. Termination happens when the ribosome runs up against a stop codon. Remember, there are three different stop codons. As the stop codon enters into the A site, there's no corresponding tRNA for this stop codon. The release factor binds in the A site. This causes the ribosome to fall apart, releasing the messenger RNA and the polypeptide chain. Let's go back to our master genetic code chart. Recall that this chart correlates codons to amino acids. When you want to find out what amino acid is specified by a codon, you find the first letter of the codon on the left side of the chart. The second letter is found across the top of the chart, and the third letter is found down the right-hand side. Some of the amino acids are specified by more than one codon. For example, proline is specified whenever the codon starts with CC. It doesn't really matter what the third nucleotide is. A, U, C, or G will all give you proline. This is called the wobble effect. Now it's your turn. Pause the video and try to find the amino acid that is specified by the codon GCC. You should have found that GCC codes for alanine. We will work more on this in class. This process is not perfect. Occasionally, changes in the genetic material will happen. These changes are called mutations. The normal DNA, without any mutations, is called wild type. There are two kinds of mutations: substitution mutations, in which one nucleotide is substituted for another one, and insertion or deletion mutations, in which one or more nucleotides are mistakenly added or deleted. When you change one nucleotide with another, for example, you have an A instead of a G, it can result in three outcomes. Sometimes there is no effect on the amino acid coded for. This is called a silent mutation. The genome has changed, but the proteome has not. If you exchange a nucleotide and the codon now calls for a different amino acid, that is called a missense mutation. Depending on which change was made, the protein may still be functional. If the substitution causes the codon to code for a stop codon, The mutation is called a nonsense mutation. During translation, the ribosome will encounter this new stop codon and stop translating. The protein will be cut short because it has an inappropriate stop codon in it. Depending on where it is, this may or may not result in a completely non-functional protein. Another type of mutation are insertion or deletion mutations. These can be devastating to the protein. Normally, the ribosome will start at AUG and then read the messenger RNA in 3 nucleotide blocks all the way to the stop codon. However, when nucleotides are added or deleted from the DNA, it causes a shift in the reading frame. Let's see how this works using real words that we are familiar with instead of codons. The reading frame of messenger RNA is determined by the start codon, AUG. Look at the letters on the screen. Take a moment to stop the video and see what you will get if you break these letters into codons starting with AUG. You should have been able to read "The big dog ate the red car." Let's say you have a deletion mutation in the DNA, and after transcription the T in the second codon is lost. The ribosome will start at the start codon and will continue to read in 3-letter codons, but the reading frame has shifted. With the T missing, all of the words are completely different. Let's pretend you have an addition mutation, but this time it's further away, or what we call downstream, from the start codon. Now, some of the initial words are okay, but after that addition mutation, the remaining words are shifted. This is exactly what happens in a protein. A change in the reading frame will result in a change of all the amino acids downstream of the mutation. An insertion or deletion mutation in the DNA almost always results in a non-functional protein. Sometimes you can have a deletion of 3 codons exactly. While this doesn't necessarily cause a frame shift, you're going to be missing an entire amino acid that can, depending on the primary structure of the normal protein, cause incorrect folding of the protein. That's it for today. See you in class.